Cleavage is defined as the rapid, repeated mitotic division of a zygote, all taking place inside a rigid, enclosing shell. This sequence is governed by a strict timeline. The entire process completes exactly 6 days after fertilization. During these 6 days, the overall volume of the zygote remains static. This creates a biological paradox where cells divide rapidly despite the total structure failing to grow. Since the total volume is locked, each division creates an increasing number of progressively smaller cells. These individual units are known as blastomeres. The structure enforcing this constraint is the zona pellucida. It serves as an inflexible boundary, keeping the embryo's diameter absolutely locked as blastomeres multiply. Yet, despite this rigid barrier, early nutrition is transported directly through the zona pellucida. This mechanical hollowing solves a major physiological problem. A newly fertilized ovum contains a massive volume of cytoplasm, far too much for a single nucleus to manage efficiently. This 6-day sequence operates as a mechanical downsizing algorithm, partitioning that massive volume into manageable units before the embryo attempts to attach to the uterus. Essentially, cleavage is a solution to a mathematical constraint. The original zygote must subdivide its internal contents into functional pieces without exceeding the rigid limits of its protective shell. The first structural milestone occurs when the zygote partitions into 2 cells, one large and one slightly smaller. Divisions follow in rapid succession, with the 2 cells quickly splitting to form 3 and then 4 blastomeres. At this early stage, these individual blastomeres are loosely arranged and remain physically distinct from one another within the shell. The third round of division brings the embryo to the 8-cell stage. As the notes here indicate, this exact stage marks the beginning of a crucial morphological shift called compaction. During compaction, the 8 spherical blastomeres change their physical shape. They begin to adhere tightly to one another to maximize their surface contact. This squeezing effect seals the outer surface and forces some cells to become entirely enclosed by the others. Note the physical transition from these early, loose, distinct spheres to this newly formed, tightly packed, integrated structural unit. Compaction represents the embryo's first act of spatial organization, fundamentally changing its internal geometry to prepare for future cell lineages. By the 4th division, the structure has reached 16 cells. This 16-cell stage is formally known as the morula. The name is derived from its appearance. The densely packed, bumpy exterior resembles a mulberry. Looking at this morula stage in the diagrams, we see the cells are still strictly confined within the original diameter of the zona pellucida. Because of the previous compaction, 2 distinct populations have emerged, an inner core of cells and an outer layer. This establishes a critical spatial rule. The outer mass of cells is now destined to form the trophoblast. Conversely, the inner mass of cells will form the embryoblast. To understand why this relentless subdivision without growth is necessary, we have to look at the structural result. The goal is to correct the massive cytoplasmic imbalance found in the original zygote. As listed here in the principal effects of cleavage, the outcome is the restoration of the nuclear cytoplasmic ratio. By day 4, the individual blastomeres have been downsized enough to match the volume of standard somatic cells. The biological math is now balanced. The cells are the correct size, and their spatial position has sealed their future tissue destiny. Around the 5th day, the cleaving embryo completes its travel through the fallopian tube and reaches the uterus. At this stage, a new mechanical force appears. External fluid from the uterine cavity begins passing through the outer layer and into the morula. This diagram shows the fluid accumulating, hollowing out a pocket and displacing the inner cell mass to one pole of the structure. This newly formed internal fluid cavity is the blastocele. Cavitation changes the architecture of the embryo. It is no longer a solid ball, but has acquired the shape of a cyst. This 32 to 64 cell structure is now formally classified as a blastocyst. Within the blastocyst, the outer trophoblast layer differentiates into 2 subtypes based on its proximity to the embryoblast. The section of trophoblast cells in direct contact with the inner cell mass is the polar trophoblast. The remaining cells that line the empty blastocele cavity are the mural trophoblast. We can see the distinction here. The polar cells touch the inner mass, while the mural cells form the rest of the outer lining. This influx of fluid serves as the final mechanical trigger, hollowing out the structure to define the points of contact required for uterine implantation. If we compare the day 1 zygote with the day 6 blastocyst, we find their total outer diameters are identical. Over 5 rounds of division, the cell count has multiplied, yet not a single micrometer of overall growth has occurred. The massive initial cytoplasm has been successfully partitioned into dozens of functional blastomeres. The nuclear cytoplasmic ratio is now fully restored, allowing each of these new cells to operate normally. The first major structural achievement of this process is the isolation of the embryoblast, the precursor to the human body. The second is the creation of a specialized trophoblast lining, designed specifically to interface with maternal tissue. This entire spatial differentiation was driven by the physical positioning of cells during the compaction stage. This entire morphological transformation was completed in just 6 days. The resulting structure is now a highly pressurized, fluid-filled cyst. By hollowing its interior and partitioning its volume, the embryo has transformed into a specialized vessel ready for uterine implantation.